Good afternoon, thank you for joining us. This is the 7th stream class of the Bioinformatics workshop organized by HiDucator and NUIG. We have worked through already several topics during the past days: databases, simple bioinformatics tools, and most recently working with 3D protein structures. I hope that you enjoyed the Chimera demos yesterday. In my opinion, those protein models look awesome. In the upcoming classes, we turn our attention to the next major topic, which is how to use Ensembl as a resource for information on genome-level data. This is again a huge field, and we see that using the covered resources can enhance a lot molecular biology research. So let's dive in.
Good morning again. We will be changing topics again from databases of single protein entries and protein structures. We are moving into full genomes. One of the main goals in this course is to identify human variants, protein coding variants in genomic context, and the best tool I can think of for that purpose is Ensembl genome browser. So today we'll be learning basics of the genome browser, and tomorrow we'll move into variants. Now the slides I have are modified from a recent set of slides used in teaching of Ensembl run workshops. They are the big experts for their tools and for teaching them, and they are provided with a liberal license, so I can use them and modify them as long as Ensembl is credited for their creations. And this E exclamation point symbol at the bottom is for indicating that this is Ensembl material. Now, in this first set of slides, we talk about what is Ensembl, how is data imported there, and we'll look at how to navigate in the website. We'll look at the help sources and get some idea of what genes and transcripts are in Ensembl and how they are presented. So what's the big idea of genome browsers? We could just have a printed book with 100 volumes, and that represents the human genome. Well, obviously that's not very usable. So the first genome that was sequenced was just 5 kilobases. You could print it on a couple of sheets of paper and maybe do some meaningful work just by cutting and pasting pieces of paper with a glue stick and setting them next to each other. Doing translations, looking at the translation table and pen and paper to get the amino acids from the nucleotides. But we really want computers nowadays and versatile tools for looking at large genomes like the human genome, 3 gigabases. And so this slide kind of answers the question in the slide. The sequence itself is not really worth anything, so we need more information to bring the data into knowledge. And these genome assemblies we have in Ensembl for hundreds of vertebrate species are really good platforms for importing all possible gene annotation, automated annotation from different sources to give a unified interface into looking at these positional annotations. Variants are really easy to see in the genomic context, and then we have comparative genomics with so many genomes, many of them very similar, like between all vertebrates. So doing the comparisons and building ortholog tables and paralog tables, we get a nice evolutionary view of many protein families. There are also gene trees which enable us to pull ready-made alignments that were used in creating those gene trees. There's a wealth of regulatory data, but unfortunately we don't have time to look at the exciting parts of comparative genomics or regulation in this 2 hours that has been assigned for Ensembl related features in this course. Now, the main content in Ensembl might be the gene builds, trying to annotate automatically every possible genome, bringing in ready-made annotations done by research groups, but also doing a consistent level of annotation based on all data. We'll see how that works in a couple of slides. If you have your own data, you can also upload it into Ensembl and use the same genome browser environment to put your data into a genomic context and to see if the variants you discovered are already known Ensembl variants, etc. There's data analysis for variants. Most of all, probably of most interest of you might be the variant effect predictor, VEP. You can export data in fairly large scale to pull, for instance, all the promoter regions, 1000 bases before the transcription start site of every human transcript. And that's in use for you in a couple of minutes. You can do programming. There are application program interfaces that allow you to do programmatic access to Ensembl data. And all of this is open source, so if you want to have a shadow Ensembl in your garage where you are secretly sequencing the dinosaur remnants that your friends have been digging, and by some miracle they've discovered some intact DNA and you have the sequence and you want to do it with Ensembl, so you are free to do that. You can install the software and have it running. There are more than 200 genomes even in the main Ensembl, which has a focus to vertebrates, and here's a phylogenetic tree of the species. The main Ensembl also has a few reference species for comparison, which are not vertebrates. There's D. melanogaster, the fruit fly, and C. elegans, the nematode worm, and some, I think, sea squirts. So the main Ensembl is just vertebrate genomes, but there's a parallel site, Ensembl genomes, which is actually 5 parallel sites dedicated to different groups of the tree of life. So if you're into insects, you would be going for the metazoa, where we have hundreds of metazoan genomes, including 24 species of Drosophila. The Ensembl is renewed every few months, but between releases the data is stable. They can't do incremental updates as in UniProt, so that they would be adding single sequences, single new proteins into it, but this is such a monolithic system that you have to do a complete rebuild. And what happens is that they add new genome assemblies, do software updates, etc., build the comparative genomic tables, bring in new data on variants, on regulation, etc. And then finally, if Ensembl 102 was released in November last year, so Ensembl 103, the current one, is there just a few weeks ago or a few days ago. And now the genome assemblies which we have in Ensembl, I think this is a pretty good metaphor for a genome assembly. So if we have Earth as a true real-life object, in the same way genome, DNA within a cell is something that clearly has a sequence. But the representation we can build of it is not complete. We can't do infinitely deep sequencing to have the full sequence of one cell and no gaps in the genome, etc. So it is an incomplete representation of the genome. And then there comes the question of which cell. So we could have an infinite number of different cells to sequence different individuals, and you have to decide on a reference version of the genome to which the individuals would have various differences. And so this map of the Earth is like a primitive representation of the actual Earth, in the same way that the genome assembly is a representation of a real genome. But anyway, it provides a coordinate system. We can number the chromosomes, and it gives us a way to place any positional annotation, protein coding genes, variants, regulatory sites, etc. And in other kind of an approach, you can think of the genome assembly as the assembled sequence reads. You align them and use complicated math to decide on the optimal and most correct alignment once you've discovered the overlaps between the fragments, and then the assembled sequence is the genome assembly. Well, actually this is just a contig sequence which will be then further assembled into the genome assembly. So when we have bacterial artificial chromosomes that contain pieces of DNA from various donors, individuals who donated their DNA for the Human Genome Project, these bags have been distributed to labs all over the world to have them sequenced. And then the contigs from various individuals are assembled. There might be overlaps again, and you get a scaffold, a run of contigs with no gaps. And then the scaffolds will then be assembled to chromosomes with gaps for the regions that couldn't be sequenced, like the centromeric region, which is highly repetitive. And if we have genetic maps and markers which we can identify in these contigs, so that will allow us to tell in which chromosome and in which arm and which cytobands these contigs were. And now the letters in the contig names we still see today are actually coming from real initials of the donors in the Genome Project, so that's a nice nod in their direction. For many species, there are alternative genome assemblies, in particular for human, which is biologically and medically such an important species, at least for us. So every now and then, when new data has accumulated so that some gaps in the genome have been closed or some misassemblies corrected, so there are new versions. This happens every couple of years. And so if we go back 2 versions, the Human Genome 18 was 150,000 gaps, and in the 37 there are still quite a few gaps, and many of them have been closed in Human Genome 38. Now, this is an improvement, but it's also an annoyance. So if you have analysis results, sequencing read mappings or whatever that relies on genomic coordinates. So if the coordinates are changing, you will be totally off in your results. A variant which was in a coding region of a gene might be in an intergenic region after the coordinates have shifted by 20,000, which might happen. So this is something that people do carefully and only every now and then. But anyway, this Human Genome 38 is the current one, and you should be using that, but be aware that you might be getting data which has been mapped in the years in the past, so that they perhaps have the human genome 19 version numbering. And in a similar way, for many organisms, there are past assemblies and more recent ones with more data. But data of all these, or information of these assemblies, is available in Ensembl documentation, so you'll find out which exact version is being in use and when it was completed and so on. And some statistics of the assembly as well. Now, as our first hands-on, we'll look at the Ensembl homepage and just a list of genomes, and we'll look at dog genomes and try to find if there are multiple breeds, which breeds have been used for sequencing, and then we'll take a look at one of them and try to find the coverage of that genome. Coverage is simply the excess of data that has been sequenced compared to the genome length, and to get a chromosome level assembly you need probably at least 10 times the genome length of data. If you have just twice the genome length, there will be many places where you have no data at all, and it will be very much in small pieces. But modern genomes are more like dozens of times covered, so a 30 times coverage is considered reliable for assembly. Here we are on the Ensembl homepage. We have information of the recent release, and we have information of various functions we could learn here, and we have a list of species here. And what we'll look at first is their help facility. So if you want to learn more, the help and documentation is really well prepared, and if we look at tutorials, they have lots of short videos that help you get started. And if you really need to use Ensembl, or let's say the variation tools in Ensembl for real work, so you might want to go to the specialized videos, and there are even full courses. So Introduction to Browsing Codate Genomes, a 3-hour tutorial, or a browser workshop, 8 hours of lectures. So you can really study in a more serious fashion. So this is just a scratch on the surface, which we'll be able to do here in a couple of short demos and some homework. Now let's go to the list of genomes and see what kind of information we find if we want to discover properties of a genome. Here we have the species. They are in alphabetic order by the common name, and we wanted to find dog. You see that there are lots of birds and fishes, and if you knew all of these fish names and bird names previously, I would say that you are not telling exact truth there. A bit up, and now here the very first dog to be sequenced was a boxer, and then of more recent ones we have Great Dane and Basenji, and in case you didn't notice, my teaching assistant Gaudi is a Basenji dog. So we'll take a look at the statistics of the Basenji genome. So karyotype and statistics. The nice thing in having multiple genomes of dog breeds is that there's also dog-specific variant data, so that they show the breed variants as SNPs. And now here's the summary of the Basenji genome assembly. So the annotation is pretty nice, 20,000 protein coding genes. It's more or less the same amount for every species of vertebrates. And I'm trying to find the information of the coverage. I wonder if the link to the assembly would give us more information. No. And even here, there's a more recent version available. Okay, but we get some interesting information here. The dog was called Wags, and we have information of the parents, African imports. And here we have information of the instrument. It was with Pacific Biosciences long-read sequencing machine to a coverage of about 45 times. And let's say if we have information of the N50, the length above which we have 50% of the sequence, of all sequence. 3.1 MB for the M50, which is reasonable. But anyway, let's not go further. We found what we wanted, a link to the assembly information, and also the fact that Ensembl could update to the assembly version 0.2. Okay, this is enough of the genomes, and we can move back to our... As the next thing, we'll start looking at genomic locations in the Ensembl genome browser. One of the important features in any genome browser is scaling, so that you can look at different levels of compression. You can look at the content of entire chromosomes. You can look at chromosomal regions. You can look at a single gene locus. You can look at a single exon. You can even zoom in all the way, so that you'll see sequence bases. But how to do that? The best way is to do a hands-on, so that we go to a region in the human genome and see how this works. And I'll be presenting some parts of the screen and what different things mean, what to look for on a location page. We are back on Ensembl homepage, ready to start exploring one genome in human genome in region in human genome in detail. And I clicked on human in the favorite genomes, and I copy-paste the genomic region. The notation is such that we have a chromosome number, colon, starting base dash ending base. And go, we should be jumping to this particular region. Now, the region in detail view comes in many different scales on top of each other. Here on the top we have a ideogram of the chromosome 4, which lists some assembly exceptions. We might look at them in more detail, but we are not going to do it now. And then this red box here represents the area that has been enlarged in the view below, and this view below is a region containing a few genes, and then the bottom section is actually the region we were looking for, and this middle section I think is chosen to be 10 times the width of the smallest region in detail. We could change these views by manipulating these marquees, so we could select a region and jump to this region, but anyway, I'll skip it. We'll just use the region we have. So closing this jump to region. And here in the most detailed view, what we have here is a blue line which represents the genome, the name of the contig sequence which you find in the sequence database like GenBank is in the middle. And the annotation is on both sides so that we have genes on the forward strand which are above the genome blue line, and you can recognize them being on the forward strand by having the arrowhead pointing to the right. And then we have genes of the reverse strand which are below the blue line, and here the arrowheads are pointing to the left, so they are on the reverse complement, the minus strand, and they would be read from right to left here. And this is important if you want to figure out where is the beginning of the gene in the graphics. So on the reverse strand the gene begins here, whereas on the forward strand the reading is the same as in European printed books with text starting from the left. Or let's say books with the European alphabets. So we have the chromosome zooming into a region which has a number of different genes, and again here we don't have any separation of the forward and reverse strand genes, but you just have to look at the arrowheads to see on which strand they are. And then we have some information of the gene quality, like the gold genes, which are most reliable, but we'll talk about genes and transcripts in a minute. Now you just want to see the navigation from chromosome to smaller regions, and you can see that in this medium region view there are multiple contigs with alternating dark and light bars here. We have the coordinates of chromosome 4 here, and you can see that there's a different scale. This is 1 megabase. That's actually the criterion. They are showing 1 megabase environment, not 10-folds time. 10-folds the amount. It's close now. This is 78 kilobases. Okay, we have lots of other data here. We have human cDNAs, which are like primary evidence for the presence of genes. We have ESTs, which are low confidence short sequence reads, not of the high throughput kind, but the technology we had 10 to 20 years ago. And we have an abbreviated regulatory build. We have some variant data here. So it's really dense, and we have the explanations for the colors, so you could start looking at the regulatory build colors and see what the different things mean. The red thing here is a promoter and so on. We'll talk about variants in more detail in tomorrow's lecture. But this is the region in detail. And we have different views here in the left, and the toolbar on the left is changing depending on where we are. So currently we only have the location tab and we don't have that many items here. But if we jump into a gene, we could do it by just clicking on a gene name here, for instance. Most things are links here in the diagrams. If you see your cursor being a finger, it's a link. So this would allow us to jump into gene information of this NUDIX hydrolase 6. So this would be a jump to the gene page. The pages are customizable so that we can configure the page and select things we want to see and not want to see. There's a huge amount of data items, but again, you'll go crazy if you just try to figure out on your first visit all the possibilities there are. So if you just change the options and click on the tick mark on the top right, so that will save and use your configuration. There is a reset configuration somewhere here. Reset configuration that will bring us back to the default in case you did something you don't like and can't remember what you changed. But anyway, this is region in detail. Today we have 2 files of lecture slides. We just finished the Ensembl introduction and move on to Ensembl genes and transcripts to look at more details of individual gene loci. Ensembl takes its annotation from 2 main sources. One is the automatic annotation they create themselves, which works for all the species. And then they have manual annotation from the Havana project for a few selected species. There's human, rat, mouse, and zebrafish. The automated pipeline, which will be able to understand at least superficially after 15 minutes, that's something that creates a lot of annotation, bringing in systematically positional information for lots of external data sources. And some of the mapping is just mapping of existing experimental data, but then we also have a pipeline of predictions to create annotations, best guesses based on other species. The biological evidence in the automatic pipeline includes the nucleotide sequence databases to have all the cDNAs, coding DNAs, presumably for proteins, ESTs, these short expressed sequence tags, which are fragments of mRNAs, usually 5' or 3' terminal. And nowadays we have RNA-seq data also, coming from the 3 members of the International Nucleotide Sequence Consortium. Then we can also map proteins to be more sure of protein coding regions, taking the UniProt content as the main data source. And then when we can't map data from the sequence itself, we can start doing the predictions. And for instance, we have little data of the chimpanzee, but with the knowledge of chimpanzee protein coding genes being 98.5% identical to human, it's quite trivial to map the human cDNAs and proteins from the human genome onto the chimpanzee genome, and thus create 20,000 protein coding genes annotated in the chimpanzee genome fairly reliably. And the further away you go in the distance of species, the more hypothetical it's going and the sparser the annotation becomes. But the RNA-seq data is increasing really quickly, high throughput sequencing to bring information of expressed genes in so many more experimental species. But in case of the manual annotation, they can take into account much more data. So they can look at RNA-seq databases, look at reliability data, try to find long-read data of the transcriptomes, use cage data, which is labeling of 5' ends of mRNAs to get reliable annotation of 5' ends, the actual starting points of transcription. There's poly-A-seq data, which would be identifying the starting point of polyadenylation for mRNA, and that would be then the end point of a transcript. And the human annotators can also do critical reading of publications to try to figure out which of alternative annotations is correct. And obviously this is very data intensive, expensive and slow, but then we do need high quality data, so that's been done for a few select vertebrate species. And so the obvious benefits are getting more comprehensive annotation so that we have reliable gene annotation for more genes than just the automated pipeline could produce. And so the number of genes and transcript is increasing due to manual annotation. And you don't need to have such huge amounts of sequence data when the human curator can decide on uncertain looking cases based on the expertise. But this is definitely slower and it is in small scale, so the information is only increasing in a linear fashion proportional to the number of people hired to do this annotation. Whereas the pipelines can be scaled up pretty drastically just by buying more computers to do that and buying more sequencing machinery to create more sequence data. Now, when we have the transcript diagrams in Ensembl, so they are given as lines including these bars, and if you guess that they might be exons and introns, you are absolutely correct. Exons, protein coding parts of the genome, are, or actually transcribed parts of the genome, spliced and exported regions of the genome are these bars, and introns, the regions which are spliced out and left in the nucleus, are in the thin lines. And we have non-coding parts of an exon in white and then coding parts in color. So we can assume that we have a stop codon right here where gold is changing into white. Now in these golden transcripts, the annotation of the manual Havana project and automatic annotation of Ensembl are in agreement, so they can be taken with even higher confidence, and they are like the gold standard of transcripts. That's probably why they want to picture them in the gold color. You might have multiple golden transcripts for a single gene if there are alternatively spliced versions, both of which have been confirmed, or all of which have been confirmed. Then we have something which is called the GenCode set of transcripts or genes. And this is important because this is the background data set which is used in many huge bioinformatics projects like the GNOMAD variant project, the 1000 genomes, individual sequencing and variant project, and others. And in the GenCode, they make a merging of the Ensembl automatically annotated genes and Havana manually annotated genes. So we have the gold transcripts, and we have all the ones which are only known to Ensembl or known to Havana. And the membership in the GenCode gene set is indicated on the page, and that's a good piece of information when you are evaluating the transcripts. Then a very recent thing from a couple of years ago is the main transcript matched annotation from NCBI and EBI. And this means that we have 100% identity even in the non-coding regions between transcripts in these two major databases. And current version of the main select database, version 0.91, includes about 90% of human protein coding transcript. So this is really good progress. Previously we had CCDS, conserved coding sequences, in which a similar comparison and agreement that this is the official version was only made for the coding regions in transcripts. But now with more technologies to get the transcript ends right, so they've been moving into mapping complete transcripts into a higher category of reliability. And so with the previous knowledge of different classifications of transcripts, so it should be fairly easy to make sense of the transcript table. This is a transcript table of one gene in Ensembl, and we have multiple transcripts here, one per line. And now we have first knowledge of, in the color, what type of a transcript it is, whether it's a protein coding golden transcript or a protein coding other transcript, or in blue it would be a non-coding transcript. There would be no protein because of a retained intron in case of this one labeled 204. And then we have all these flags. This is an important region in the transcript table which indicate what all projects have approved this particular transcript. So you can see that the top one is in the main select, so that would be probably a very good one. And now if you look at other information, if you try to make a decision between the top two, so you can see that the conserved coding sequence is the same, so they code exactly for the same protein, which you can also see from the same UniProt code and the same length of the protein for both of them. But there is a tiny difference in the non-coding length between these two. 2274, 2283. But if I needed to care about the non-coding sequences, so I would just trust the main select process and choose the top one here. And then this APRIS is a project which tries to map alternatively spliced transcripts into principal and alternative isoforms. And this is done by comparison of the protein forms in different species. It is assumed that the isoform of a protein which is conserved between species would be the principal one, and the alternative splicing which brings variants, might be more like species specific. There might also be expression data to show that one form is expressed everywhere and the alternative form is just expressed in specific tissues. But here we can see that these top two versions are labeled apris principal, and the 2 is a lower quality indicator, probably because there are 2 of them and they can't make a clear selection of one top transcript. And then we have the alternative version, another protein coding sequence which produces a different protein with a different CCDS. And then for some proteins we have flags of coding sequence being incomplete in the 5' end or in the 3' end, and obviously these are not good to use, and they might be just mistakes in the splicing system or just incomplete data from whichever sequencing project. But for our purposes we can easily ignore them and only focus on the ones which have golden transcripts and the main select and apris labels and possibly CCDS labels and refseq labels. So this top one is certainly voted by all instances to be the most reliable transcript, and this is the one we could use. And furthermore, there's transcript support level, so they have scores 1 to 5, 1 being best, which is ranked by having data available. So TSL1 means that there is sequencing data to support this transcript, and for others there might be just fewer transcripts available, so these get lower TSL scores here. So really a lot of information in just a few kinds of flags. But when we go to the page of a gene to see this transcript table, there's help available, so mousing over on these flags will give an explanation what they are about to remind you. Okay, these colors are also available in the gene views. This is an example of transcripts which have been shown aligned with a region of the genome. So we can see that for the BRCA2 gene we have this gold transcript, which is the most reliable one. We have an additional one which has some minor difference. Maybe it's in the end here. It's also protein coding and might produce the same or a slightly different protein, but anyway, it's ranked to be not as good as the golden merged transcript, which is both in Havana in Ensembl projects. And then we have a number of non-coding transcripts in blue. And so the coding exons and introns are labeled with the bars and lines, and the open bars are for non-coding parts of an exon. Sometimes it could be an entire exon. Now, Ensembl objects are labeled with stable IDs so that the accession numbers are not changing, which is pretty good. But if corrections are made so they don't create a new random accession number, so that we would need to look at some history table to see what was the old version. But instead there is the stable accession ID and then period and a version number. So 0.1, 0.2 here are references to what is the version number of that particular Ensembl gene, transcript or protein or exon or regulatory feature. So The ends here is an indication which helps when you have a database code out of context. So seeing the 3 letters ends help you immediately, you know that, okay, this is Ensembl data. And then the next letter, if this is human data, so this tells you what type of an object it is, whether it's gene or transcript or peptide or what. And if we have other species, not human, so there's an additional 3 letters after ends but before the letter for the object. So for mouse it is mus, taken from the Latin name, and in case of zebrafish, Daniareria, there are 2 letters from the genus and 1 letter from the species name. So you can do a lot of inferences from just seeing an Ensembl code before you put it into Ensembl for searching. And obviously, as soon as you know it's from Ensembl, you can copy-paste, open Ensembl and do an Ensembl search to get more information. And the things here marked with hashes would be just a unique identifier number for that object in that species. Now let's do a short hands-on and go to see a gene page and try to make the evaluations of the transcript we have there, and let's see what other information we can find through the links from a gene page and a transcript page. We are on the front page of human genome and we want to search for a specific gene, and so I'll paste, and now I could have gone to the drop down on the left and select a category to search like gene, but it seems that we already have a direct link to a human gene here, which is given as the most probable search target, so we can just go there. And now this is a gene page, and you may see that there's a lot of more data items here on the left. And the transcript table is not shown by default, so you need to click Show Transcript Table. And once you do that, it will be showing like forever. And now I need to scroll a little bit to see all of the information, and maybe I'll decrease the font a little bit. So here we have only 3 protein coding transcripts, and just one of them is labeled with the gold color, and all of these have the same conserved coding sequence, the same UniProt code and same length, so these code for identical proteins, and all of these represent the principal isoform with great certainty. So it's probably a difference only in the non-coding parts in the ends of the transcripts. So there's a difference of more than 1,000 bases between the shortest and the longest. But we would be choosing the main select, and the mouse over I advertised is coming here. Now if we want to see information of a single transcript, we don't have a transcript tab at the top. So we have just the location and the gene tab. So clicking on the transcript will open us a new page, and technically it's a new tab here on the Ensembl page. So the gene page is still there, so we could jump between the gene and the transcript just by clicking on the top of the tab. And now for the transcript we have some data that wasn't there. We have information of the of the exons and a diagram of this transcript, and also the data items here have changed, so we can go and retrieve the cDNA, which would be just the exons together from this link, or we could find the protein sequence from here. And now this is pretty ascetic compared to even FASTA. This is just the sequence, no annotation. But the interesting information is coded sequences and alternating exons. So black is the first exon, blue is the second exon, and then they would be alternating black blue if there are more. And now the red letter here indicates that this glutamine is encoded by a codon which overlaps the splice site. All right, we don't really have time for the zebrafish problem presented in the last slide, but that is something that I would be doing and presenting in a forum posting for your entertainment.